Hi, my name is Brian Kim, Professor and Vice Chair of Research in Dermatology, Director of the Mark Lovell Center for Neuroinflammation and Sensation at Icahn School of Medicine at Mount Sinai. Diseases associated with type 2 inflammations, such as atopic dermatitis, prurigo nodularis, chronic spontaneous urticaria, asthma, eosinophilic esophagitis, and food allergy affect millions of people worldwide. You may wonder how type 2 inflammation underlies the pathology of these considerably different diseases. In this video series, we'll review 3 dermatological diseases associated with type 2 inflammation to highlight how dysregulation of distinct cellular and molecular components of the immune system imprint unique responses even within the same organ system. These basic principles can then be applied to understand how type 2 inflammation can also play a role in diseases across various organs and systems. There are 3 basic immune modules and corresponding inflammatory pathways that evolve to defend against different kinds of pathogens. While type 1 immunity protects against intracellular pathogens like bacteria, protozoa, and viruses, and type 3 immunity is specialized to combat extracellular bacteria and fungi, type 2 immunity protects against parasitic helminths. When any of these responses go awry, they may result in a number of different autoimmune or allergic conditions. Although many diseases can have overlapping immune axes, underlying their pathogenesis, the predominance of particular modules is what enables targeted therapeutic approaches, for example, blocking TNF-alpha in psoriasis. Cells involved in the type 2 immune response are recruited by stressed or damaged tissues, and when activated, produce and release large amounts of vasoactive mediators, cytokines, and proteases for expulsion of parasites, wound healing, and neutralization of toxins. Dysregulated type 2 immunity can also lead to allergic and other associated diseases. A number of innate and adaptive immune cells may be involved in type 2 inflammation. In general, innate immune cells are poised to respond rapidly to tissue cues, while adaptive immune cells are more specific in their ability to generate a tailored response to an antigen. Let's take a closer look at these different cell types and how they come together in type 2 inflammation. T helper type 2, or Th2 cells, which differentiate from naive Th0 cells, play a primary role in the adaptive type 2 immune response. Th2 cells are key drivers in establishing and maintaining immunologic memory, and therefore can drive chronic inflammation. Classically, antigen-presenting cells migrate to the draining lymph node from the tissue, for example, the skin, and present the antigens to naive T cells to become Th2 cells, which are characterized by the production of IL-4, IL-5, and IL-13. These cytokines can then activate other adaptive immune cells, such as B cells in the lymph node, to activate the humoral immune response. When activated, B cells differentiate into plasma cells. They produce several classes of antibodies, of which IgG is the most abundant. Similar to Th2 cells, which recognize antigens, B cells are considered part of the humoral arm of the adaptive immune system due to their ability to coordinate an antigen-specific response via antibodies. In the setting of type 2 inflammation, many plasma cells switch production to IgE, which is normally found in very small amounts. Elevation of IgE is a hallmark feature of many allergic disorders and often underlies type 2 inflammation. Indeed, Th2 cells, particularly via IL-4, stimulate B cells to produce antigen-specific IgE, which binds to high-affinity receptors on mast cells and basophils. More on this in a bit. Group 2 innate lymphoid cells, or ILC2 cells, are a population of tissue-resident innate immune cells that closely resemble Th2 cells and produce large quantities of IL-5 and IL-13. However, they are starkly different from T cells in that they lack antigen-specific receptors. Activated by epithelial cell-derived alarmin cytokines, ILC2s are poised to rapidly respond to barrier breach and amplify the type 2 immune response. Mast cells are widely distributed tissue-resident granulocytes that are primarily located in proximity to blood vessels, sensory neurons, and at barrier surfaces. Classically, mast cells, although innate in identity, can become activated in response to antigen because they bind IgE at the cell surface via their high affinity receptor, Fc epsilon receptor. IgE cross-linking degranulates mast cells, resulting in release of factors like histamine, but also the release of lipid mediators like leukotrienes, and even production of type 2 cytokines like IL-13. Basophils, also granulocytes, share similarities with mast cells, including expression of the high-affinity Fc epsilon receptor, but are largely found circulating in the blood. They migrate into stressed or damaged tissues and are a major source of IL-4. It is increasingly appreciated that both mast cells and basophils also become activated independent of IgE. In addition to mast cells and basophils, eosinophils are another type 2 granulocyte that respond potently to IL-5. In fact, ILC2s are often a major source of IL-5 in recruiting eosinophils into tissues, where they release various molecules and enzymes that contribute to tissue repair or allergic inflammation. Historically, type 2 inflammation was believed to be driven by adaptive immune responses primarily involving Th2 cells. And indeed, Th2 cells play a critical role in all type 2 inflammatory diseases and the generation of IgE. However, we now appreciate that a variety of cell types, including a host of innate immune cells, are also major sources of a variety of factors that promote type 2 inflammation. In type 2 inflammatory diseases, certain cells likely play larger roles than others, helping to explain the specific features and clinical manifestations of each disease.